Welcome to NOAA Central Library's platform for the presentation of ideas, research, and news in support of the National Oceanic and Atmospheric Administration's mission. Today's library seminar, titled Sustainable Use and Stewardship of Ocean and Coastal Resources, is the third in a 5-part series sponsored by the NOAA Research and Development Enterprise Committee, focusing on the 2020 NOAA Science Report. Fisheries representative of the committee, Dr. Michael Liddell, will introduce today's 5 speakers. Before I hand the mic to Mike, here are a few logistical tips to help you enjoy our presentation. If you're having trouble with the audio or visual components of GoToWebinar, please log out and rejoin us. This resets the software and resolves most technical issues. Our presentation is being recorded and will be available on the NOAA Central Library YouTube channel in a few hours. We want to hear your questions. Please feel free to type them in the questions chat box located in the control panel. However, let us know to whom the question is addressed by indicating the name of the person you would like to answer your question. Questions will be answered at the end of all of the presentations. So with that last detail, let's get started. The mic's all yours, Mike.
Hello, and welcome to this webinar on sustainable use and stewardship of ocean and coastal resources, the third of 5 seminars in the NOAA Science Report seminar series. The next seminar in the series will be held on March 24th at 3 p.m. I am Michael Liddell. I am the Fisheries Representative to the NOAA Research and Development Enterprise Committee, a cross-line office committee of the NOAA Science Council. This committee is responsible for the development of the NOAA Science Report with input from contributors across NOAA. The sponsors for this seminar series include the Research and Development Enterprise Committee and the NOAA Central Library. The Science Report is an annual report that highlights scientific accomplishments for the year. The Science Report celebrates research and development in 4 sections. There's an introduction, a science highlights section, a section on our publication bibliometrics, and a section focusing on NOAA's scientific workforce. Together, these sections highlight how NOAA's research products impact the lives of all Americans. The 2020 NOAA Science Report will be released later this month on the NOAA Science Council website. The report spans the entire range of NOAA's mission. There are more than 60 stories featured in the 2020 report, stories that represent the accomplishments of NOAA employees. Today, we welcome speakers whose work centers on sustainable use and stewardship of our ocean and coastal resources. The bios for the speakers today can be found on the calendar and website description of today's webinar, so I'll just give the names and affiliations of our speakers in order to save more time for their presentations. Our speakers are Dr. Mark Rowe, a research physical scientist at the Great Lakes Environmental Research Lab. Steve Kibler, an oceanographer from NOAA's Beaufort laboratory. Dr. Matthew Loretta, a research fishery biologist at the Southeast Fishery Science Center, and Jason Spires, a research ecologist at the NOAA Cooperative Oxford Laboratory, and Chris Holderried, director of the NOAA Catchmac Bay Laboratory. Each of the speakers will give a brief presentation and we'll have a question and answer period at the end. So, Mark Rowe, you're up next.
Thanks, Michael. And I think my slides are already showing here. So I have a lightning talk on the Lake Erie harmful algal bloom forecast and new advancements that were made in 2020, which included transition to operations of the new forecast model, as well as I'll highlight a few publications results. And I want to give credit to a number of collaborators on this work over the past 4 years or so, from the NOAA Great Lakes Environmental Research Lab, NOAA National Ocean Service, and the Cooperative Institute of Great Lakes Research. And I also want to acknowledge the Great Lakes Restoration Initiative, which was an important source of funding for this work. So there was an incident in 2014 known as the Toledo water crisis, so algal toxins were detected in the finished drinking water at the city of Toledo, which resulted in an emergency water shutdown. that left 400,000 residents without drinking water for 3 days. And there were some economic estimates associated with these types of events. The economic cost of a severe harmful algal bloom season just in western Lake Erie is about $65 to $70 million. And just in terms of consumables for drinking water treatment, the cost is about $4 million a year. And the city of Toledo itself spent about $500 million to upgrade their water treatment infrastructure in response to this incident that occurred. But these economic estimates, they don't really get at the heart of the total cost of the actual emergency shutdown. And this article on the right here is showing that there's been a lasting effect of this emergency shutdown, that a lot of residents still never regained confidence in the drinking water and continue to buy bottled water, which is a significant ongoing expense to this community. So if we can predict events like this and help drinking water plants be prepared and help them to avoid these types of emergency shutdown events, that can be a great benefit to coastal communities. So this figure is showing the components of this forecast that we developed and transitioned to operations. So this building on the Lake Erie operational forecast system, which is an operational system run by NOAA that was new in 2016. It has a new high resolution model based on FBCom, and it predicts three-dimensional currents in Lake Erie. And then there's also a component of satellite remote sensing. There's a product called the Cyanobacterial Index, which shows the location of the harmful algal blooms. This product is produced by National Ocean Service. And these products feed into this Lagrangian particle model, which is a new model that we put together for this updated forecast. And that predicts the three-dimensional movement of the bloom over the next 5 days or so. And we added a component to this model to predict the vertical distribution of the cyanobacteria in the water column. And the result of this is predicting the location and movement of the bloom over the next 5 days. So a new component of this model was the ability to predict when the bloom forms surface scums as opposed to when it's mixed down through the column. So the buoyant cyanobacteria, they float up to the surface during calm conditions and they mix down to the bottom during windy conditions. So recreational users are concerned about when these scums form, because that's where the highest concentrations of toxins can occur. Whereas the drinking water plants draw water from the bottom. So they're interested in knowing the concentrations at the bottom. So this type of model can produce both of those types of predictions. So in addition to that vertical distribution, the updated model had better predictions of the horizontal transport of the bloom. And this is important because that water crisis that I mentioned was associated with a transport event that flushed the toxins out of Maumee Bay and moved it along the coast past their drinking water intake. So the 3D model was better able to predict the distribution of the HABs over time and avoided this artifact that you can see in the bottom lower panel where the bloom tend to pull away from the coast, which is something that was not observed. That was an artifact of the pre-existing model that we were able to improve with the updated model. And in 2020, NOAA GLERL worked with NOAA co-ops to transition this model to operations, and they also developed an updated website to present the information to the public with updated graphical presentation, which you can see here. And the final thing that I'll mention is an ongoing research item. So we're building on this operational Lake Erie HAB forecast into a new product that incorporates data from the weekly monitoring program that NOAA GLERL and SIGLR conduct, where we measure chlorophyll and toxin concentrations in the lake. And then we developed this probabilistic model to predict the probability of exceeding the public health advisory toxin level in the lake. And so the animation you can see on the right would be the final product that we would show to the public, where we can see the probability of exceeding this public health advisory level for the toxins. So that's a product that would be of interest to both recreational users of Lake Erie as well as the drinking water plants. And so this is in a research mode now, but we're working toward developing a public product out of this. So that's a brief summary of this work, and I'll hand it back to Michael.
All right, thank you very much. Next up is Steve.
Thank you very much. what I wanted to talk to you about, just a little bit of information about some forecasting tools we're developing, molecular tools to aid forecasting in the Gulf of Maine and other parts of North America. So a little bit of background information. We're targeting Alexandrium. The species is Alexandrium catenella, which is a planktonic toxic dinoflagellate. Forms blooms all over the northern half of North America, as shown on the map. The harmful effects of this species, it produces a suite of toxins. Once they get into shellfish that are consumed by people, the end result can be paralytic shellfish poisoning, severe illness, potentially fatal in some cases. And as the name suggests, one of its hallmarks is paralysis of mussels. In addition to the human health impacts, there's also resource problems with shellfish, shellfishing bed closures, and limitations to shellfish aquaculture. One of the interesting facets about Alexandrium catenella is its ability to form resting cysts, which are a dormant stage that overwinter in the sediment, germinate in the springtime, producing the spring bloom of Alexandrium cells. And after a few decades of research, it was determined that Alexandrium cyst abundance can be a good predictor of bloom intensity in the water column. So because of this, we can use the abundance of Alexandrium cysts as a forecasting tool for the spring bloom. And currently there is a forecasting model that is running for the Gulf of Maine. It is initialized with the wintertime concentrations of Alexandrium cysts. Now, the method that cyst abundance is determined it really involves a lot of work to get to this point. The current method is a microscopy-based method, which involves, in addition to collecting samples at a grid of 53 stations across the Gulf of Maine, is a series of processing steps, including sonication, sieving, chemical preservation, and staining with a fluorescent dye called primulin. The end result is shown in the two images here, where on the left-hand side, you have a light micrograph showing a cyst right there in the center, and then the corresponding fluorescent image of the cyst after it's been stained. Some of the problems with this method, it's very time-consuming and labor-intensive. There's a great deal of training and skill that's required to identify the Alexandrium cysts and to differentiate them from those of other species. And the end result is a lot of variability among scientists, both in terms of their different equipment they use and some human factors. The whole method is a little bit subjective. And finally, this produces a lot of variability in cyst abundance estimates, which can affect the forecast. So because of this, we turn to some molecular methods and specifically genetic markers to try to improve this process. Because we could use these methods for unambiguous cyst identification, remove some of the human factors, and speed up the whole process. So the two methods that we turn to, the first one is called Qpcr Quantitative Pcr. It's a method that really estimates the number of copies of a specific gene within the target cells. And as you can see on the upper left-hand side, it's a plot of Alexandrium abundance in cells per liter versus the number of copies of this gene. And this relationship can then be used to predict or to estimate the number of cells that are in the sample. And that's shown on the lower panel where we have Alexandrium cell abundance in cells per liter at a site between 2012 and 2017. The second method that we focused on was called FISH, or fluorescent in situ hybridization. The name is long, but it just really means there's a fluorescent dye that is attached to a specific genetic sequence in the target cells. And you can see the results of this in the upper row of this series of figures, where on the upper left-hand side, you have Alexandrium cells in a clump. To the right of that is a fluorescent image using the species-specific FISH label, and then the overlay of the two is on the right-hand side. The other three rows are the different types of controls that are required to perform the assay. A side-by-side comparison of the two methods, Qpcr uses a DNA primer, which targets a gene sequence in the ribosomal genes of the target dinoflagellates. The end result is the number of gene copies and then the conversion to the number of cells that's equivalent to that. In terms of comparison, FISH we used a PNA probe, which is an artificial DNA. Works just the same as regular DNA, it's just more stable and penetrates into the cysts better. Again, it targets a specific gene sequence in the ribosomes, but this time attaches a fluorescent dye so that they can be identified with microscopy. For pros and cons, for Qpcr to the left, it's rapid and high throughput, extremely sensitive, and there are commercial standards available to make cross-laboratory validation possible. Some of the cons are the precision is only moderate, really to about a half order of magnitude as good as it gets, but as it turns out, this is good enough for the forecast models. It requires a Qpcr machine and a molecular lab to pull it off, and it really hinges on knowing the number of copies per cell, which can be pretty difficult to figure out. For fish, you can identify cysts unambiguously with the fluorescent label. The fluorescence can be tailored to avoid background fluorescence from, say, chlorophyll or other pigments, and multiple labels are possible for different targets all at the same time in different colors. Some of the cons are it requires fluorescent microscopy gear. The microscopy can be fairly expensive. You still need a lot of lengthy microscope time for actually counting and identifying the cells, and there's a lot of controls required in order to make sure this works properly. In terms of the status of the project, it's still at a relatively early phase. We've got a lot of validation work still ahead of us in order to start moving this toward having a useful product to help the forecast. We're starting with the Gulf of Maine because we've been doing forecasting there for years already. But the intention is to speed up the process, make it more available to users, and to use it to do forecasting in Puget Sound, California, and places like Alaska. And that gives a quick summary of the project and where we are, and I appreciate the time.
Thank you very much to both of you. That's a very interesting pair of talks highlighting two very different methods for modeling and forecasting HABs, a hydrodynamic approach and a genomic approach. So it's very fascinating and highlights the breadth of NOAA's work. Next, we're going to move to Matthew Loretta, who will give us another application of genomics.
Hello, everyone. I'm really happy to be here today to tell you about some of our recent advancements in the genetic assessments of Atlantic bluefin tuna. This is a project that is a multinational collaboration with collaborators that span the globe, and I'm happy to be here to represent some of our outstanding work that we've accomplished in the last couple of years. As a basic introduction, first thing to know is let's define what is close kin mark recapture. It's a powerful technique that's been advanced lately for both human populations and animal populations. It's a genetic approach to identifying individuals in that population using the DNA profiles of their closely related family members. For example, parent-offspring matches, siblings, or half-siblings. You can think of this as an ancestry type database for fish. The technique can be applied to natural populations to estimate the total abundance of reproducing individuals. It's based on two fundamental laws of nature. The first is that each individual contains the genotypes of their parents, one male, one female. And the second is that individuals are highly related when the breeding population is small and vice versa. In an extreme example, if there was only 2 breeding individuals in a population, then all the offspring would be full siblings. And if there were 4, for example, and breeding was random, then you would have 25% unrelated, 50% half siblings, and 25% siblings. And that's the basic concept, that as the breeding population grows much larger, the relatedness of their offspring becomes much more diluted. And we like to use this principle to solve two major problems with the assessment of tuna in the West Atlantic. The one is to identify the stock structure of fish caught by fisheries. We know that the stocks mix extensively in the West Atlantic with 2 major breeding grounds in the Gulf of Mexico and Mediterranean Sea, as well as other areas that larvae are being found throughout the Atlantic. But this mixing causes problems for estimating abundance by stock of origin. However, using this technique to where we can identify individual stocks and match them to offspring in known spawning areas, we hope to estimate the absolute abundance of the fish that are spawning in the Gulf of Mexico annually. Let me dive a little deeper into the methods here on how this is done. Essentially, the study design looks at DNA samples from fish caught up in the northeast US and Canada from our fisheries, which are primary hand lines and rod and reels. These fish are genetically compared to the larval fish collected in the northern Gulf of Mexico from earlier that year, where spawning peaks in approximately April and May. Then by estimating the ratio of matched mature fish to the young of the year, using these parent-offspring pair matches, essentially the equivalent of a fish paternity-maternity test, it's possible to estimate the total number of spawners that are spawning in the Gulf of Mexico. One of the biggest challenges in designing a mark recapture study is to ensure that the population of fish that you are marking then mix with the unmarked population such that your recapture event is representative of those fish that were marked and the others in the population that weren't marked. Here we're looking at how we are able to validate and design our study based on some of the current information coming in from satellite tagged fish in the West Atlantic. Here we're looking at fish that have been identified as entering a known spawning ground, whether it's the Gulf of Mexico, and then here are shown in blue as Western stock. And these sort of polygons on the map show you their spatial densities in a distribution. The red are the ones that were observed entering the Mediterranean while they were being electronically monitored. These are satellite tags and archival tags that track the GPS locations of fish when they're attached. And then the green are the fish that weren't observed entering a spawning ground. But essentially what we see is that the Gulf of Mexico component of the population seems to start its migration events in around November from the northeast fishery area, entering the Gulf of Mexico as early as December, but really piling up on the northern spawning grounds in April and May. Now, NOAA has a larval monitoring program in the northern Gulf, which covers that entire region during May. So here we collect plankton samples that contain bluefin larvae, and we wind up collecting those and genotyping those to mark fish that are actively spawning in the system that year. Then by June, by the end of June, fish have migrated out of the system back to the northeast, and we can see throughout July, August, September, and October that they're well mixed with the other stocks. And by sampling this as a recapture event, we hope to get a representative sample of fish using the West Atlantic that we can then compare to our marking event. So here's just a little overview. If we look at the figure on the right, the upper one shows the standardized grid of the larval cruise that goes out each May and samples the northern Gulf. And the gray circles show the approximate locations of fish. These are the ones from 2016. Then in 2017, we tried a different approach where where larvae were found, positive sites, we did this heavy aggregation sampling to see if we could increase the numbers. And you can see that, in fact, we were able to do that successfully so that we can now have an approach to gain lots of marks as part of a supplemental sampling to our standardized cruise. What we found from our pilot study was that within those first two collections, we genotyped approximately 850 fish. We found that over 700 unique spawner genotypes were contained in those larvae, successfully genotyped out. We found that some larvae were related, but that overall the relatedness within a larval sample was low. This is important because the fish are captured within the first week or two of their life, such that there was no guarantee that they might be from different spawning pairs. But we found that on average, they were fairly unrelated. In an ideal, perfectly mixed situation, you would get 2 spawners for every marked larvae. Here we saw a slight reduction at 1.6, but still a pretty good return on the sampling effort. Then when we looked at some of our fishery catches from Canada and US from 2017-2018, we actually found several parental matches with larvae collected earlier that May. This provided the proof of concept that we could then increase our sampling efforts to use this approach to get a robust mark recapture estimate of the Western stock, as well as the composition in our fisheries to other stock to give us an overall good picture of the total number of bluefin tuna. Interestingly enough, we also found 2 half-sibling pairs in the Gulf of Mexico between these years. This is the first genetic evidence of spawning site fidelity to the Gulf of Mexico from one year to the next, and also opens up an exciting possibility of monitoring spawner activity in the Gulf without ever having to handle the large mature fish. So the next step for us is that we have this large scale marking event that occurred in 2018 where we took this aggregation larval approach and we collected over 3,500 larvae. This is exciting in that we think now we can use this to mark thousands of fish in the system beginning in that year and then track them over time in the fishery catches through over the next few years. And then the other component of this is looking at the contribution of other stocks to our fisheries. This is important as we define the recapture sample. We want to make sure we're pairing the larvae to the appropriate stock, that is the ones that match the Gulf of Mexico. So here on the left figure, you'll see a general overview of our current fishery surveys that have been collecting DNA over the last few years. And the table on the right shows the sample sizes by fishery year. That's the total fish genotype so far. Those that associate with the larvae from the Gulf of Mexico as being of the same population structure, those that identify with the Mediterranean larvae as being of the same population structure, and those that have their identification is uncertain. And if overall we look at the percentages, we see quite a large contribution of the Gulf stock, of the Mediterranean stock to the West Atlantic. A couple patterns to note here are that one, as you go more northernly, the fish appear to be, in the fisheries appear to be a higher component of Gulf of Mexico origin than as if you go to the Gulf of Maine or even more south to the large pelagic survey. And two is the temporal trend over time, that the population seems to be increasing its proportion of Gulf of Mexico origin stock as we get to the recent period. So this is a first comprehensive look at the composition of the fish, the stock of origin to the fisheries. And you can see the sample sizes are quite large. I'm sorry, I'm not, there we go. So the conclusions and next steps from our pilot studies. Close-kin mark recapture integrates all biological sampling programs for Atlantic bluefin tuna. And that's an exciting thought that we now have a method that links these different surveys into a comprehensive program in this ancestry type database building. Two, we learned that larvae provide effective large-scale genetic marking of spawners on the spawning grounds. This is good news because these fish are active in the system at the time of marking, and they migrate for recapture with only a few weeks, yet have a well mixed. So this is a dynamic unique to bluefin tuna that we can use to our advantage in a mark recapture study. Second, that the fisheries effectively sample the mixed adult populations. So far, we've genotyped over 6,000 fish from the West Atlantic, and our goal is to get to 10,000 fish by this summer. And from this, we hope to get these information products within the next 2 years that have been proven hard to get by traditional assessment methods. The first is the time series of stock composition in the West Atlantic fisheries. The second is the mark recapture abundance estimates of the Gulf of Mexico spawning stock. And then the third is to get the true fecundity. That is, when we build enough parent offspring matches, we should be able to find out exactly which fish spawn in the Gulf of Mexico. That includes what stock of origin, what size classes, and age classes. All these have been difficult to get in the past. So with that, I would just like again to emphasize that this is a large effort amongst researchers all over the world, and I thank them all for their time and effort to this to sort of advance the bluefin tuna science to the next stage in a very robust and exciting way. Thank you.
Thank you very much. This time we're going to move on to Jason Spires from the NOAA Oxford Lab.
All right, thank you very much. Thanks everyone for your interest, and thanks for joining us giving us the opportunity to share our work with you about testing alternative oyster restoration strategies. I want to acknowledge the primary PIs on the applied portion of this work, our primary co-PIs at the NOAA Restoration Center, the United States Naval Academy, Maryland Department of Natural Resources, and Hampton University. The species of interest for this work is the eastern oyster. Chrysostrea virginica is a keystone species in coastal ecosystems, providing valuable ecosystem services to many of these regions. But in these regions, the population levels are depressed. So there are large-scale oyster restorations efforts underway, as well as a ongoing aquaculture industry. There is wild harvest persisting. So what this talk is going to share with you is the development of a tool, of a marking tool that we think could benefit many of these sectors that I just mentioned, and then the application of this tool to test an alternative oyster restoration method with the NOAA Restoration Center. So some of the specific stakeholder needs that an oyster marking tool could fulfill would be to study growth analyses, or to deter theft from aquaculture farms, or the monitoring of stocked individuals, or understanding of population connectivity, specifically in the larval phase. And then this last bullet, we use the tool that we developed that I'll talk to you about to test an alternative setting method. So techniques for marking fish are well developed, and here's a number of images of tools that you'll be familiar with for marking different species. If you focus in on the bottom right here, you'll see this fish glowing green that's been tagged with a chemical marking tool. And that chemical is called calcine, and that's the drug that we have been working with, and that we're going to go over a few method slides to develop a way and test a way to mark different life stages of the eastern oyster. Some of the benefits to chemical tagging are that it can be very safe and efficient. If you look at this image in the bottom right here, you can see a researcher at the U.S. Fish and Wildlife Service marking thousands of fish fry at the same time. A little bit about oyster biology before we dive into this. Oysters use dissolved compounds like calcium to create their shells through biomineralization. So the drug we're working with, calcine, binds with calcium in the water. Oysters take in those minerals for new shell formation, and that shell that was bound with the fluorochrome dye excites under the right light source. The chemical is a non-toxic, non-hazardous fluorochrome compound, and we have to work with the FDA and U.S. Fish and Wildlife's Investigational New Animal Drug Approval Partnership Program, who allows researchers to mark specific life stages of different species. So there are two objectives for our work. The initial methods piece was really to determine the efficacy of the fluorochrome dye calcine to produce external markings on different life stages of the eastern oyster. So we tested late stage larvae, which are called petiveligers, juvenile oysters, and adults. And then we took what we learned from that methods investigation and applied it, working directly with the NOAA Restoration Center, to develop and test additional methods for setting oyster larvae in a Chesapeake Bay tributary. So we released calcine tagged oyster larvae, and we attempted to recover tagged juveniles. And if we did find juveniles, we wanted to quantify how efficient were we, in that if we release X number of larvae, how many spat did we recover from that release? So the results from our methods piece is we learned that oysters can be marked, and all life stages of oysters can be marked successfully. The eastern oyster tolerates exposure to calcine with little to no effect on growth or mortality. You can create multiple marks on juveniles. That's the 3 rings that you see here in this upper left image. Marking the youngest juveniles multiple times creates the most distinguishable labeling, kind of like barcoding. And that the labeling persists in individuals that were labeled as juveniles for over 3.5 years post-mark. And then if you look along the right-hand side, marked larvae, which is what tile F is, carry that mark through metamorphosis. So when they transition from being planktonic to sessile and they become a juvenile, you can see the mark in the juvenile shell as they grow. So I said there's an applied piece to this, and we were working with the NOAA Restoration Center to test an alternative oyster setting strategy. Well, what's the normal strategy for restoring oysters? How that works is it's called remote setting. You spawn oysters in captivity, you put their progeny in tanks, you feed them algae. Then when the larvae are old enough, you move them to tanks and you add lots of settlement substrate. In most cases, it's oyster shell. And the larvae attach to that shell, and then you transport that shell to the reef, the restoration site. Well, why the Restoration Center is interested in this piece is sometimes the limiting factor in the restoration work can be the shell component. So we wanted to see if we could reduce some of the reliance on that shell and test a strategy that removes putting larvae in tanks with settlement substrate. And in this case, you take the developed oyster larvae, which is here in the palm of your hand, millions of individuals, and you take them to the reef of interest for restoration. And in our case, we release them with divers. And we also use the chemical tagging tool that we've been developing to tag these oyster larvae so that if we recover juveniles, we would know their larval origin, because we're conducting this work without any enclosures in space where natural recruitment is occurring at the same time in Chesapeake Bay. So how we did that was we released, or we created our own artificial oyster reefs because we wanted to be able to sample thoroughly. So in this case, we created an oyster reef out of shell bags that we deployed with our research vessels, and that we could recover at the end of our larval releases 8 days later to sample to look for juveniles. We released larvae at these known locations within the reef in about 15 feet of water, and we cued the larvae to strike with some natural chemical stimulants. And like I said, we released the larvae with divers directly on the shell, and then picked up the shell 8 days later after we gave them time to attach, to settle on the shell, and then transition, metamorphose to juveniles. Here's what we found. Many spat juveniles were found exactly where we released them. X marks the spot. If you look at each one, at this figure on the right, each one of these cells is a shell bag, and we had replicate reefs constructed. And if you look at the cells with the X in them, you'll see these have the highest number in them. We found thousands of individuals right where we released the larvae. Overall, our setting efficiency was about 1%. And we were also monitoring current speeds at the time of larval releases because we wanted to reduce the planktonic drift. We hit slack tide deployment, which was a goal of ours. So we were trying to get the larvae to stay right where we released them. Here's where the tagging comes in. There was no evidence of natural recruitment. Each spat that we found, each juvenile, contained the calcine tag when we looked at them under the right lighting conditions. This means that these juvenile that were growing were from the larvae that we released. There was no natural recruitment mixing into our experimental restoration reefs. So our next steps and additional collaborations are to test this over a larger scale. You know, we did this on very small controlled research reefs. So this summer, we're hoping to work within the NOAA Choptank River habitat focus area to test releasing larvae on intact oyster communities via a boat with a towed deployment mechanism that trails over the reef in order to determine the viability of this alternative method for large-scale restoration activities. In addition to that project, we're also working with other collaborators to better understand population connectivity of oyster communities in and around marine protected areas in other systems. So thank you all very much for your time and interest, and I want to say thank you very much to the COL staff who's helped out for the last 5 years with this work, and acknowledge our external partners.
Thank you very much. Our last talk for today is Chris Holderreed.
Thanks, Michael. Really appreciate the other talks I've heard, and between the fish and the halves and the oysters, you're going to hear a little bit about that in my talk, just from the up in the Northwest in Alaska. So I'm going to talk about a project we have to do an ecological assessment of Catchmac Bay, and really want to acknowledge a huge team of folks working on this from our NCOS labs, from different labs really around the country, here in our Oxford and Silver Spring offices in Maryland, as well as Beaufort lab in North Carolina, and a whole bunch of partners regionally here that are helping us put these products together. So I want to start to explain the motivation for the project. Really want to take you on a little tour of Catchmac Bay, and including Homer, which is shown here. You know, the communities in the bay depend on marine resources. So whether that's fishing, tourism, maritime shipping, marine trades, and a lot of subsistence food harvest. And you know, these resources are include really important commercial and recreational fishing, clamming, ecotourism, which is growing, oyster farming, and other mariculture, which is also growing. And to support all of these industries in the changing environments we have, we really need, you know, they need good information about those changes. And that's one of the things that we're trying to address. And, you know, there's a rich diversity of animals, birds, invertebrates here, as well as the harvested species. So from protected resources as well as harvested species, we really have a lot within the bay. I mean, I have to say it's a spectacular place to live and work. And all of that is supported by the habitat. So we have a very diverse range of marine habitats, from kelp to seagrass to mudflats, salt marsh. We have a whole diversity of different glacial watersheds, and a big rocky intertidal ecosystem as well. But the challenge is that we have this very rich system that has also seen a lot of decline. So we've had crashes in crab and clam populations, had a whole series of really strong marine heat waves recently that caused crashes in fish populations. We've had increasing harmful algal blooms, and there's a lot of oil and gas development in Cook Inlet, but there's also expanding mariculture. I'd mentioned that before. So a lot of rich resources and a lot of challenges for those resources. And that one of the advantages that we have in the bay is that there's actually a lot of organizations here, between both on the NOAA side, state agency side, tribal partners, really a whole range of folks, marine conservation, folks working in the bay, collecting information, doing monitoring and surveys and things like that. So there's actually a lot of information available. The difficulty is much of it is not readily available. If it's available, it's actually not that easy to use, and very little of it was integrated together. And for all of those challenges that I talked about, really what you need is to be able to get to it, to be able to look at it in a way that's easy, and to have you know, really be able to look at more things together from the management perspective. And so our project is really about that. It's improving data access, use, and sharing of information between different organizations. Because there was not a lot of underwater habitat information available, we really had a focus on improving that, and also specifically tools, geospatial tools for shellfish, kelp management, and mariculture, so kind of newly emerging industries, and as well as trying to deal with the expanding HAB threats with better risk assessment products. So now I've kind of taken you on the close-in view of Catchmac Bay, but if we step out, just to show you where we're located. So Cook Inlet's in south central Alaska, and Catchmac Bay is on the southeast side of the inlet. And just I have to do the reference, so if you want kind of the size comparison, so we're down in south central, that's the yellow circles around Cook Inlet. If I then compare the size of Cook Inlet to Chesapeake Bay, they're about the same size. Cook Inlet's quite a bit deeper. And then Catchmac Bay is about 40 miles long, has a max depth of around 600 feet. So for Alaska, it's relatively shallow, but it's deeper than most of the East Coast locations. And then one of the things that is a particular feature for the bay is that we have 15 glaciers feeding from an ice field on the south side of the bay. that empty in with this whole diversity of different watersheds. And that's one of the things that produces the rich systems that we have in the bay. And all those habitats that I mentioned earlier, they are representative of the larger Gulf of Alaska. So the bay kind of becomes this natural laboratory that is a microcosm for what we find out in the Gulf. And because we have all these organizations working here, and the bay is a state designated critical habitat area, it's a national estuarine research reserve, our Kibler Bay laboratory is there. It also was designated by NOAA as a habitat focus area. So from those programs, we actually have a lot of information to get at some of the challenges that we're facing. And just a sense for what that is, this is, you can pick out the agencies that might be interesting, or the topic that you might be particularly interested in, but we really have a lot of information here. So it makes it a particularly good place to integrate information to better address Alaska coastal challenges that I mentioned before. And it's one of the better places in Alaska to be able to do this because of the combination of what the natural system is and the information and agencies that we have in the area. So what we're doing is basically producing a whole suite of different information tools to inform both resource managers, the public, tourists, and a whole range of folks, fishermen, charter operators, just across the board in a range of different ways. So some of it is data. It's pulling together information, telling people where it is or making it available directly. Some of it is providing access to that data. So you're actually making viewers where you don't have to be a GIS expert to be able to look at the data. You can go online and see it. And then in other parts, it's looking at publications and reports. So actually summarizing that information in ways, both in technical scientific manuscripts, but also in reports that give people a snapshot of what's going on. And that's actually one of the things we did in 2020 was to do a state of Catchmac Bay report, a pilot report, to kind of have the summary of the status of the bay right now. And so I'll give you some examples of several of the products that we've put together recently. So one of them is the pilot state of the Catchmac Bay report. And the way we did this, we took advantage of all the partners in the area, so many contributors, and we wanted to keep it engaging and flexible. So we had a lot of different content and also had it represent what the particular agencies wanted to show. So whether that was monitoring data, whether that was new techniques that they were using, we had flexibility to do that. And then the goals were just let people know what's going on, give us a tool to track changes over time, share information between multiple stakeholders as well as the agencies, and then hopefully develop a pilot that can be transferred to other areas in Alaska as well. And then just giving you a sense of what some of this looks like. It's a 10-page document, range of different things from monitoring data to snapshots of information to historical information, as well as kind of trying to put things in context between the heat wave and how that affected fisheries and harmful algal blooms, for example. So another way I'd mention the mapping and visualization tools, and NCOS has produced a Catchmac Bay Biomapper. So it's an online GIS visualization tool that combines things like here we have the benthic habitat map for Catchmac Bay, as well as all those dots are where we have drop camera videos. And so there's videos and photos there that you can access. And also combined with other habitat mapping information, as well as like the green in there where the seagrass beds were identified. So it just easily puts together a lot of habitat related information for the bay. Another approach is to, more on the educational side, is looking at story maps. And that's combining both visual and mapping information, and in this case, both recent and historical information to look at the very large changes in sea otters over the past 20 years, as well as how those changes in the population are affecting marine ecosystems. And it's something new that's coming out. So we've had the 2D biomapper for a while, but brand new, which is going to be this coming year, is a 3-dimensional biomapper. And in a place where you have, 3,000-4,000 foot mountains and very interesting bathymetry, it really gets a better look at what the ecosystem gives everyone a better look of takes you into the ecosystem is probably the best way to say it. So then our next steps really going forward this year is we're looking to incorporate the feedback that we're getting from the partners into the 2020 State of the Bay report, and then have that be an annual product. The 3D biomapper I mentioned will be coming out. We're also pulling together an ecological status report, and that's a longer report that's looking at the changes over the past 20 years, essentially since the Catchmac Bay National Estuarine Research Reserve was designated. And they did a large characterization at that time, and then this will be an update since then. Also, I'd mentioned pulling together the geospatial data to make that more available, and then taking the integration of some of this information to develop habitat suitability tools, particularly for shellfish management. So again, just cycling back to really our goal, it's giving our communities the tools they need to deal with those challenges, whether that's resource management, kind of the new mariculture opportunities, the dealing with the risk of oil spills and harmful algal blooms as well. But one of the things we really realized in the process of doing this is it really helped us with information sharing among the agencies, and that we're looking forward to that being kind of a more routine way in which we work. And I can't, I almost can't, since I have the last talk, I'll bring up the Google Meet, in our case, not the Zoom window. I just have to really appreciate the large group of people across, from my NCOS colleagues who have been working on this project, and especially Karen DeCino up here and Gretchen Messick at Oxford for really making it all work. So, thanks.
Thank you very much. I want to say thank you to all of our presenters today. It was a really wonderful collection of talks. I was really struck by how great a showcase it is of NOAA's work. We had talks that ran from the Gulf of Mexico to the Great Lakes, from Chesapeake Bay over to Catchmac Bay. It covered the whole breadth of the country, and it covered a variety of techniques from basic technique development that we had from Jason and Matthew talking about, to development of operational products that we had Mark talking about and Chris talking about. So it was talking about the whole breadth of NOAA's research enterprise coming together to basically address a single issue of coastal resource management across the country. So I want to thank everybody. It was an excellent showcase for the work that NOAA does. At this time, I would invite people to ask some questions. We have just a couple minutes. I believe you can type your questions into the chat and we will present them to our speakers.
Yes, hi, this is Lisa from the library, and I just want to reiterate that if you have a question, we can take the next 6 minutes to answer them. Just make sure to let us know who you're addressing when you ask your question in the chat box. And while we're waiting for those questions, I also wanted to bring your attention to the handouts drop-down menu that's in your control panel. A few of our presenters have graciously added their presentations as a handout. So if you're interested, take a look and feel free to download them in the next few minutes. And one last thing before I read this first question, I'd like to encourage you to share this webinar with interested colleagues. You could find the link to the recording on the NOAA Central Library YouTube channel later today. I have a question. It says, for Dr. Kibler, can the molecular techniques you described be used for HAB species aside from Alexandrium?
This is Steve Kibler. Yes, the short answer is yes. They're fairly general. Our project was really to tailor them for Alexandrium cysts. These methods are used pretty widely for a lot of species, including a variety of HABs.
Thank you. And thank you for that question as well. I'm going to keep encouraging people to ask them while we have our speakers available to us. Again, I wanted to also thank you for your lightning talks. I was really impressed and very interested in the work. I can't wait till the report comes out. Okay. I'm going to wait just a few more seconds to account for the delay that we often have between asking a question and seeing the question.
While we're waiting, I'll take a quick moment to plug our next seminar. 2 weeks from now on March 24th, the 4th seminar in the NOAA Science Report series will be a robust and effective research, development, and transition enterprise. The seminar will include lightning talks about innovations with the geostationary lightning mapper, next generation satellite products that monitor coral bleaching and heat stress, the development of next generation models for seasonal to decadal predictions, and the updates to geomagnetism models. So another diverse set of talks in 2 weeks.
Excellent. We did get 2 more questions. This first question is for Dr. Holdery. When compiling the data from so many sources, how do you account for varied spatial scales or data quality considerations when looking at the ecosystem scale?
Yeah, that's a very good question. And a lot of it, in fact, the one where that came up probably the most was on sea stars, because we have sea star monitoring that's done through our Gulf Watch Alaska program. So it's done on a very, it's done in a way with the sampling that lets you do inferences over a large geographic area. We also have citizen science volunteers that are out there doing, sometimes daily when they're going out to the Peterson Bay Field Station, but much more frequent measurements, but at particular sites. And I'll tell you, it was a very long discussion about how we combine those different types of information. And part of it was realizing that you need both, right? But you have to be a little bit cautious about how to combine them. So I'll just say that you just have to think through it. Our bias is that we want to use more information, and that we've been talking, we have the Catchmac Bay Science Conference coming up next week. And one of the themes there is that we're all doing science. So whether you're a researcher or a citizen scientist or a fisherman out there who's been observing things every day on the bay, we really want to bring all that information together. So thank you for the question.
Thank you for the answer. I appreciate it. This last question is for Jason. It asks, are any oyster farmers using the marking techniques you showed, or would they?
Not yet. Not yet. So we're currently in the investigational stages of using the mark, working with the FDA and U.S. Fish and Wildlife to provide them data on the efficacy of the marking tool. Once they gather enough data from us and other researchers, the FDA could then grant the use of the drug, because it is something you're adding to an animal that can be consumed by humans. So right now, that farmer, if they wanted to use the tag, would have to apply with that specific program. But I'm not aware of any that are doing that yet.
Excellent. Thank you for that response. Well, that will be our last question for today. I want to conclude by thanking all of our speakers for their presentation, as well as Mike and the NOAA Research and Development Enterprise Committee for organizing this series. Again, the next episode or webinar in the series will be March 24th at 3 p.m. Eastern Standard Time. And I am very proud to say that NOAA Central Library is proud to present the work of the NOAA community, and we hope you'll join us again. So be well, everybody. Take care.